neuroscience and beyond. Know more, get inspired. My name is William Georgiev, and I'm a PhD student at the University Medical Center Göttingen in Germany. Together with my PhD colleagues, Sabina, Christina, and Lenny, we decided to bring science, and especially neuroscience, closer to the general public. In the following episodes, we're going to talk to various scientists about their inspiration to do science, their research topics, and their opinion on current scientific problems. Today, our guest is Dr. Patapia Zafeiriou. Dr. Zafeiriou is a principal investigator at the Department of Toxicology and Pharmacology at the University Medical Center Göttingen in Germany. Her research focuses mainly on the molecular and cellular mechanisms underlying neurodegeneration, infantile epilepsy, and sudden cardiac death. In order to address these questions, Dr. Zafeiriou and her group use different tools such as tissue engineering, genome editing, and stem cell technologies. Bioengineered neuronal organoids, representing the central and peripheral nervous system, are among the tools that allow her group to study the communication between neurons, but also the interaction between the brain and the heart. Before we begin, I would like to mention that this podcast is separate from my research position at the University Medical Center in Göttingen, Germany. This podcast is supported by the International Max Planck Research School for Neurosciences, as well as the Cluster of Excellence Multiscale Bioimaging in Göttingen. And now, please enjoy the talk with Dr. Zafeiriou. Hello guys, welcome. Today with us is Dr. Patapia Zafeiriou. She is a principal investigator in the Institute of Toxicology and Pharmacology at the University Medical Clinic. And today we're going to talk with her about organoids, what are these things, about stem cells, about neuroscience, and maybe about the heart. And I'm very excited, and thank you for being here.
Thank you for inviting me. It's a pleasure to be here.
Yeah. Why don't you start with a short introduction about yourself? How did you get into science? And our previous guest, Felipe Opazzo, is my colleague, and I know him personally, but we have never met, I believe, with you. And I'm also excited to hear this for the first time, your history behind the scientists that you're today.
Sure. I have to say, probably it started very early. So I really loved actually looking at bugs and little animals, and I loved microscopes. I think I bought my first microscope when I was very young. It was one of the things I really loved doing. So observing nature, I would say. And I had a very naive idea of science. So I thought, I would be probably like the scientists from 1900. I liked recording, writing stuff, and I liked pinching my fingers and looking at blood at my first very bad microscope where I could see nothing. I could tell you probably I imagined a lot of the stuff that I thought I was seeing, but I was very happy. And I really liked biosciences. I felt like that. I did study chemistry eventually with the idea to go into biochemistry, and this is what I specialized in the National and Kapodistrian University of Athens. And then I came to Germany after my master thesis to do a PhD in Charité. And then I went from Charité to Max Delbrück Zentrum. I was working with pancreatic beta cells. I came from the endocannabinoid field, and then slowly I turned more into molecular biology and cardiology at the time, because I was advised that was the time after your PhD to make a change in the field, because later on you don't really change so much. So I went to cardiology, and actually I really enjoyed it. And I came here in Göttingen in 2010, and I was still working on a cardiac project. And I went into neurons actually by chance, because as it always happens, because I had a very specific knockout model, and it was TBX5 protein, and that was a cardiomyocyte-specific knockout model. And what we saw at the time was that when we knock out this protein in mice hearts, a lot of the RNA sequencing data that we found, a lot of dysregulation was happening in the neurons. Mm-hmm. So what I did then was I thought, okay, so that means there was a lot of crosstalk of neurons and cardiomyocytes. And by the chance, my director then entered the room, and he said, would you like to do some neuronal tissue engineering? And I was like, great. Then I can somehow innervate the cardiac muscle, which is something that is generated in our labs. And then I could study this interaction also in a human setup. And I was very naive. I thought that's it. I went to brain organoid field because I wanted to study neurocardiac interactions. And I was very happy to see this through. The last years we have been working on it and building on it. And now I think we are there.
Okay, that sounds great. But can we go back to your Berlin stay? So I was also at Charité in MDC. I'm curious, with whom did you work there in these institutions?
Yes, so in Berlin, I was with Dr. Santosh Nigam. I was working with arachidonic acid products and pancreatic beta cells but I cannot even remember anymore what I really did. It was I was working on hypoxylin A3, I remember, but and Max Delbrück Zentrum. Then as I changed to the cardiology field, it was with Dr. Martin Bergmann and Laura Celarajan at the time.
I don't know them, but I was very focused on neuroscience, to be honest.
Yes, I don't come from the neuroscience field.
Exactly.
I have to say, I should have really focused on that earlier. I realized I really love it. It's so complex, but it's so fascinating. So I think it's a pity I found about it later on. I made a very long way to it, but it was fun.
Yeah, also I think it's astonishingly complex, and we have material to study for our whole life, maybe, I don't know, and it's not going to be enough even. That's why I'm so fascinated by the brain. But in Berlin, I mean, I've also been there for a couple of years, and I know it's totally different environment, big city. In Göttingen, where we are now, it's smaller. However, you have a lot of science concentrated here. How did you find this transition from the big city to the small one? Was it tough at the beginning?
Yes. I think I was pretty naive. So I came from Athens, and Athens is pretty chaotic. So when I went to Berlin, for me, it was optimal. I loved it. I really loved it. It combined some nature, relatively little traffic in comparison, and it was a very nice environment. A lot of wonderful interaction also with all the students we had there, but also a lot of things to do. And coming here, I thought it's okay. I will just go to the countryside. Actually, we found a little house close to a lake, and I thought, wow, there will be a lake, and it will be so much close to nature. And I realized I don't really like nature that much. So after living, I think, there for the first 3 years, I realized I really, it's nice to have a lot of animals around, but I realized I really miss to be actually in at least a town. So then I moved in the city. Still, this is a little town, but it's better than living outside of Göttingen. So transition was not easy. No. And I actually told my husband at the time, 2 years and we're back in Berlin. And I've been saying this the last 12 years. And I still haven't left. And I think it is because of the environment. So the scientific network here is amazing. At least this is how I experienced it. And it's very hard. It's very hard for me to leave. And I think for others as well. I hear a lot of scientists come here and are stuck here for a very long period of time.
That is true. Even I remember I applied for different graduate schools in Germany, and my professor in Berlin, he was like, okay, if you want to go to do science, go to Göttingen. If you want to combine it with social life, maybe you should stay in Berlin. So think about yourself. So I decided to come to Göttingen. Sorry?
Isn't it a little bit like Hotel California? You know, you can always come, but you can never leave. I mean, this is my feeling.
I don't know. I mean, I've been here for like 2 hours, 2 years, sorry.
2 years. Okay, then you don't know.
2 years, 2 years and a half. And for me, it's fine because I already have my social circle before I came here, and I don't feel like I need that kind of a social interaction or going to parties, theaters, whatever. I mean, I've done this in the past, and now I do it, of course, but not that often, let's say. And for me, Göttingen is a perfect balance between, okay, I have a couple of friends and I can concentrate on my work. And if I want to go party or go visit some event or, I don't know, travel, I go to Berlin or whatever. This is, by the way, one of the biggest drawbacks for me in Göttingen, the lack of airport. If I want to fly to my home country, Bulgaria, I got to go to Berlin, Hamburg or Frankfurt. Or, yeah, but still, I enjoy being here. Anyhow, as you said, there are a lot of institutions here and a lot of fun things are happening. You already mentioned cardiomyocytes, you mentioned organoid stem cells. Can you tell us more about your work? What are you doing in the lab and what are you studying and why did you choose, for example, this model organism?
So we are working mainly with engineered tissues and organoids from induced pluripotent stem cells. And
okay, I will stop you here because it's a lot of terms at once.
Yes.
Maybe we can start with the stem cells.
Okay.
What are these guys? What is induced stem cell? Can you take a cell from my skin and make it grow as a neuron, for example?
Yes, not directly. It will have to take some months, but yes. So at the end of the day, the idea is that we can take any cell nowadays, a somatic cell, what we call that normally wouldn't differentiate into another cell type. It's defined like a cell from your skin. It can also be blood. It can be from different parts. And then you can reprogram it, which means we would give the factors that define the identity of the cell. And then you could bring the cell back to an earlier time point. And this cell, then you could be able to redirect into any cell type you would like. And for us to do that, we need to give certain factors and we need to give developmental cues, what we know from in vivo studies, how actually development works, and then we can modulate the cells and then bring them to the direction we want to turn them into. So for example, if we want to make brain organoids, we need to activate the neuroectoderm commitment. If we would like to make cardiac tissues, then we have to go through the mesoderm. So there are different ways to do that by using different factors. And if you think of stem cells for us in the lab, it's a little bit like Lego. So you kind of you can make them whatever you like. By CRISPR-Cas, we can give them different colors. We can even make them cool with optogenetic stimulation. We can make them lighting. And then you have to combine them by patterning. Is it like if you have a guideline, our protocols is a guideline, how to build them together into making a building. If you make with a Lego a building, for us, it will be an engineered tissue. It can be a cardiac tissue, it can be a neuronal organoid or a neural tissue, or it can be a combination of both.
Okay, sounds easy, as you said, but I know it's not easy at all.
You can ask my PhD students.
Yes, a lot of effort and also time, as you said, and maybe problems with contamination. I don't know, maybe you can tell us more about it, but how exactly does this organoid help you study the brain, for example? You have some cells, but to what extent they resemble the real brain?
Of course. That is a question. And I think this question we are all asking ourselves. And the idea is, of course, we cannot have access to human brains. One part of us, of course, would really like to. Another part of us knows this is really wrong. So we have to have what we can have at the end of the day. And we can study these organoids. We can understand interaction between the cells and regulation. And yes, they are not brains. Of course, they're not. And if they were, we would even have ethical problems, right? So there are cells that they are together. And the idea and the magic about it is that they self-pattern. That means they self-organize and they self-pattern. It's not, if you look at it, if somebody comes from in vivo studies, they know exactly how neurons in the brain are situated. They know exactly how to cut it. For us, if you look at our organoids, someone will say, well, this is a mess. Yeah? But in reality, if you are looking at the neural network development and if you're looking at the cells, there is an organization, there is a self-patterning as well. So what we have been recently looking at is what actually the cells are doing when we don't give them anything anymore. And it is amazing what we actually discover from the moment we do not give them anything anymore. The cells that they are inside the organoids are really patterning each other. So they are really making other cells, that they make different parts of the brain.
Exactly. So we're talking about the very, very, very early development of the brain, where these stem cells differentiate themselves and grow into other cells, let's say. And we were talking about this a couple of minutes ago with our colleagues here, that there are two different concepts. One, as you said, you don't give them anything and they grow. I mean, you give them cues, but you don't specialize these cells. And the other one was, okay, I want to grow a specific region, for example, cortical or a midbrain. Do you have these methods in your lab?
Yes, so we are actually following the second method. So we are patterning them to a specific region. But what we discover is after you do that, then they have the ability to pattern into something else. So what we are doing is, for example, where the bioengineered neuronal organoid is the model we developed. And I just aimed to have a forebrain model. So I patterned the model for forebrain development. And the first 30 days, when all the factors we are adding are there, the model is really patterned for the forebrain. And the next, if we are looking then at RNA sequencing data of the subsequent days, then day 60, day 90, then we can see that slowly midbrain arises, hindbrain identity arises, and even spine identity. So, which is for me amazing, because this is not really what we intended to do. And maybe this is something that we actually have to consider. When we pattern an organoid, it is enriched, of course, in the cells we wanted it to become, but it doesn't mean that it won't then start to differentiate into something else. And this is something that, for me at least, is fascinating. It's living. So if you don't stop it, then I think it will turn and it will become more and more complex, which I love.
This is really amazing, and I mean, I didn't know about that, to be honest. But at what stage? You say that after some point, so you have to develop these things and then turn them into forebrain, and after a certain period, they start developing these different regions. For how long can you maintain this system in your lab?
Well, it really depends what you're looking at. I can tell you we had been looking at multi-electrode array. We're using the system with a lot of electrodes where we can monitor the activity of the organoids. And we had organoids there for a year, which means, of course, we are plating them on a flat surface. And we could still record activity. The activity pattern changes a lot with time. In the beginning is a lot. There are a lot of network bursts, and then it organizes in a complex network. And then slowly it decreases. We don't really know. I think there are other labs that they also suggest that you can keep them longer than a year, and it's normal to keep them so long. However, I don't think people really investigated if there are other identities. I'm sure they did, but not everyone, or at least we didn't before. Let me put it this way. We didn't do this before, and now I'm really fascinated to see really what comes up.
There's a paper, I think in Cell for a couple of years ago, where they compared this activity of organoids with fetus that were prematurely born, and there was some similarity, and they started questioning about the problem about, are these things sentient, etc., etc. I think we are far away from that, but what is your opinion about this problem?
I think we would all love out of the Frankenstein in us, to say somehow that they may be thinking, So, but what is consciousness? That is the big question. So even if we would like to prove it, what is it? So is it input? Is it output? We have to give a certain input, and then we are expecting it will have a certain output. And if we change the parameters, then it will do something else. Does it mean it's conscious? I think I cannot believe that this is the case. I think it's just an organization of cells. It's still amazing for us to understand the crosstalk of the cells and the network dynamics, and we learn a lot from them. But I do not think that there should be a concern for ethics from that point of view.
Yeah, exactly. I share the same opinion, more or less. It's. I mean, as you said, we cannot define even consciousness in humans or primates or animals, other animals, if they have it or not. And yeah, this is for me too early to tackle these problems. But as you know, in science, sometimes people love to speculate so that they get attention or not. I don't know. It's a tricky topic. But speaking of this resemblance, we know in the brain that we have neurons, we have glial cells, and these glial cells are more or less microglia, astrocytes, oligodendrocytes, and in addition to that, we have endothelial cells and vascularization. What kind of cells do you have in your system? Are they only neurons, or you have it in combination with other cell types?
No, we have excitatory neurons, inhibitory neurons, and this was also the aim of this organoid, to have a strong inhibitory network, to have also the interaction of excitatory neurons with inhibitory, which you need for network. And then we also have a lot of astrocytes, and we know that from day 80 and 90 we start getting oligodendrocytes, and our very recent and unpublished data show also some microglia. So we have also we see the rise of all the populations that you would expect coming from the neuroectoderm. Microglia normally would come from the mesoderm. And I think this points more also in combination with what I told you before, what we see is that with time development, there is a lot of self-patterning, and that we even see some spine morphogenesis, and even some, in the RNA sequencing, it is, let's say, morphogenesis of other things. So it seems like it is generating much more things. I think there is probably some mesoderm commitments happening, not from us. And this is something that we need to investigate more.
This is really amazing.
So, and there is also this idea that this microglia population, that there is a population inside in the very early brain development. And there was a paper also before that showed that there are microglia in organoids. So from that point of view, I think it can happen. We don't know really the details.
That's very interesting. I work with microglia in Berlin in MDC with Professor Kettenmann there, and they also try to differentiate. I think microglia. Don't know about organoids, but I know that they migrate, as you said, from around day 7.5, if I'm not mistaken, from the mesoderm. But it's very fascinating. I really am. I was not aware of these things. However, can you design some system that combines brain organoids with cardiomyocytes?
Yes, so this is something that was coming to the question we had in what you told me in the beginning. So this is what I actually started to do. So I wanted to have this combination, and I wanted to study arrhythmias, and I wanted to understand neurocardiomyocyte crosstalk. And for this, I thought, okay, I made a brain organoid. Now I could see what is the interaction between the two organs. I forgot one very important point, which was the autonomic nervous system. And then I was like, okay, I think we need to kind of commit the organoid more to the peripheral side. And for this, we had to pattern it. And I got an excellent PhD student that really made this happen. And he could actually generate an autonomic neuronal organoid that has sympathetic neurons and parasympathetic neurons, and we could fuse it with a cardiac muscle tissue. That is something that is established in our institute by Malte Tibursi and Wolfram Zimmermann. And once we put these tissues together, we see a functional interaction. So we can optogenetically manipulate the neurons, and we can make the cardiac muscle beat faster, which is for us proof of concept.
It's already something, a big achievement. I mean, when you say it like this. So this is one line of research in your lab, right? The interaction between the brain and the heart, or the organoids and the myocytes. What else do you study? What kind of questions actually do you address when you study these guys, these organoids?
For me, one very interesting question is actually GABAergic neurons and really their role in network development. You mentioned earlier the paper for this GABAergic, so for the network activity and the comparison with the fetal brain. I think actually this I was very interested to see that they had development of GABAergic neurons around 6 to 8 months in their organoids, and this is also where the complexity of the network appeared. We had an earlier development of GABAergic neurons, and we could see that this complexity of the network was advanced in earlier time points. So I really think that these interneurons play a huge role. And I'm quite interested in the somatostatin neurons, which are the ones that they are believed to be the hub neurons, that they are driving the giant depolarizing potentials. And this is something we are interested in, and also not only in the form of development, which is some scientific questions, but neuronal network organization and dysregulation also in epilepsies, for example. So from that point of view, and connecting that actually to the heart, you can also have that under epilepsy, some people die out of sudden cardiac death. And then you have sudden cardiac death under epilepsy, which is known as SUDEP. So I'm mainly interested in electrically excitable cell organization and dysregulation. And at the end, in the heart, that translates into arrhythmia. In the brain, it translates into epilepsy. This is, let's say, the main focus that as I'm looking at it. Of course, we have also a lot of other very exciting projects. One really exciting is actually oligodendrocytes, which are extremely important, and so we are working very hard. And by we, I mean my PhD student. I have to say this. So by we is a lot of work of a lot of brilliant young scientists, and there I have another project on the inner ear modeling and another project on midbrain modeling, and I have to say all the students are having their ups and downs, and it's a wonderful process. It's sad at some point, you suffer with them, but the moments of success are a lot of fun.
It's a good moment to change the topic in terms of PhD students. But before that, I would like to ask you another thing. I'm studying the dynamics of the extracellular matrix in the brain, and I'm interested, do you see some molecules, or have you checked at all if you have these guys in your organoids? Some kind of extracellular molecules expressed? Or you're not aware
of which specific ones? What are you interested?
The for example, the perneuronal nets, or these lecticans, or the hyaluronan, for example.
I haven't really looked at it. We can gladly look at it if you like. I haven't. So we know. I can tell you from the things we know is we have a lot of collagen that is being generated in the tissue. And I haven't really looked at so much more. So a lot of different collagens from the ones we put in, because we're generating the tissues with a hydrogel, but then you actually see they are generating a lot of hydrogel. So we get a lot of collagen generated by the astrocytes. And they also had other components. I think hyaluronic synthase was one of the things I remember, but I cannot tell you crazy details because I haven't looked at it.
Yeah, just out of curiosity, maybe you looked, I don't know. Anyhow, but the PhD students now, how do you keep them motivated? I know this is a very frustrating work. You have to come in the weekend, you have to change the medium, you have to keep them clean, etc., etc. And it's a huge struggle, as we all know. But what is your strategy to keep them going despite this frustration from time to time?
It's hard. So it's hard, but at least for me, I think the students are self-motivated. I'm very lucky that way. I think they all struggle at some point. And I think at this point, at least what I try to do is feel it, understand it, and try to tell them I'm there and we're going to go through this. If they have a technical problem, what I have to do is find someone that can help if it's not me, because I cannot master all techniques. And then I have to be able to find someone that can help them, or say, well, maybe it's not worth for you to concentrate on that and waste your time and try to do this. So direct them on how to surpass the problem. I think motivation is hard. It has to come from within. I think handling the frustration is the only thing that I can kind of help by saying, it's okay, yes, we had this and it happened and we will try it again and next time it will work. And, I think this is not so much more that you can do. Mm-hmm. I think what helps is sometimes also when the students get to go to conferences and they present their data and they're excited or they're selected for talks. I think this is, of course, it's a bit stressful also, but then it's really a satisfactory feeling. And somehow, I think they always need a little glimpse of success. And if they feel it progresses, it goes somewhere, people are interested in what I'm doing, then this keeps them staying, motivated to continue, because it's tough. It's tough. And stem cell work, it's really tough because there can be very frustrating. It's very time consuming. Every organoid experiment for most of my students is a minimum of 2 months. And that is culture. Mm-hmm. Analysis comes after that.
Mm-hmm. Mm-hmm. 2 months and it might be a failure, we don't know. So you gotta repeat it. And then, we've all been there.
Which means you run parallel experiments all the time. Mm-hmm. And you have to check that organoids work. You have to develop ways to say, okay, at this time we're looking at these markers. If it does work, then we can continue, or it makes no sense. We have to abrogate it. And of course, things can go wrong at different levels. Contaminations can happen, anything. So we are lucky with the brain organoids so far. Not so many contaminations with cardiac muscle. I think it's also not because of the tissue, but it's because you're using so many molds and so many things, then it's a bit harder. But that's the life. I think if you work with animal models, sometimes you also have to wait, and things also can go wrong. And it's okay.
A colleague of mine works with a certain mouse line, and she's waiting, I think, for 6 months. And still nothing, and it's really, really frustrating. I mean, but this is science. I mean, it's people say it's 10% success, 90% failure, and if you're lucky, then you have some success. But it's hard. But you guys are highly qualified specialists, and at some point, many of us now, especially after the PhD, they have to decide, okay, I would like to pursue a career in academia, or I would like to go to the industry. What kept you going to become a PI, for example, not going to the industry?
I always thought academia is not a choice. It's an addiction. So I think I said this also to my students before. I don't think... I always thought, yes, I will also select. And somehow, whenever the time came to select, I felt I had no choice. So maybe I will change my mind. And believe me, I thought about it almost every beginning of the week. I was like, why am I doing this? Because it's not easy. And I have 2 little kids, or not little anymore. So my daughter is 12 and my son is 8. And you kind of have to manage everything. And it is you have to manage the impossible. And it's tough. And it's, as you said, many times not as satisfactory. But if this 10%, that sometimes it's not so high, but if these moments of success make you feel that happy, as it makes me. And I think this is why, at least why I decided to stay. So the moment, I see an experiment, an idea being realized, or a hypothesis being validated, or even that is not true, and you say, okay, then we go a step further, then for me, these are the moments that keep me going. The rest is clutter.
Okay, that's inspiring, I think. But so 2 kids. This is tough. This is tough. At what stage in your scientific development were you when you got the kids? If I may ask, I don't know if this is personal.
No problem.
Feel free to not ask, not answer.
No problem. So I got my kids, both of them, during my postdoc phase, which is, I think, for me, it felt that it was not a good time. I know if you're thinking about it, probably there is never a good time. Mm-hmm. So if I could have chosen, I would have done it when I had a group, because as a character, I would have been absolutely fine to have a baby and at the same time guide someone and continue the science. Mm-hmm. And it felt to me that the moment I went to mother leave, and also I had to stay before for some time, then everything had to stop. There was nothing happening and my projects just froze for the time I was out. So I was out altogether 18 months. And I don't regret it. It was a wonderful time with both of my kids, but this time my projects froze. And a lot of things did not happen. And I think it's better if, of course, you have the chance to have a group that can continue working, or someone that can continue working. I think that would be wonderful for young postdocs. And it's typically that time where you are like finished with your PhD, and then you're like, okay, now I will have a child, and then nothing happens for that time. And this is a bit hard, because then your publication record is not the same as it could have been. And then a lot of young female scientists get demotivated and say, well, I will never be compared. I cannot reach that. So it feels like the unattainable dream.
Good that you brought that, the female scientists. I mean, we know that there is this struggle now because of these reasons and other reasons, but how did you feel back then? Did you feel, okay, this place where I'm now, Göttingen or Berlin, supports me in this regard? Or do you think we can do more so that female scientists feel more secure when they have their families, so that they can come back to science in an easier manner, so to say, without having this kind of fear, okay, now I have to take care of my child, but my project will freeze, so I will not be competitive. What do you think we should do so that we can help female scientists, also male scientists, it doesn't matter who else wants to have family, to feel somehow secure that having a family won't affect your job?
I have to say I was lucky from the point of view I didn't have pressure. So my director, he did not pressure me for the time I wanted to take, or I didn't feel the pressure of the project. I felt the pressure on my own, but it was not coming, let's say, from above. I did not have maybe support. Now looking at it, now I look at it, let's put it this way. At the time, it did not really occur to me. I did not have, as I said, something happening when I was not there. But at the end of the day, I think nowadays it looks better. I think every year, or as the time passes, it's getting a little bit better. I think now a lot of consortia are putting lump sum funds to support when a PI or member of the team is, let's say, in leave, that then they can have some funds and HIVIs. It's not a lot. It cannot substitute really an experienced postdoc. But even some sort of analysis or anything is better than nothing. Mm-hmm. So I think here in Göttingen, there are also, through MolMed and so on, you have chances to have financed over heavy when also young students are going out in pregnancy and in mother leaves. So I think from that point of view, it feels it's getting better. And probably I was also not informed enough. Maybe that would be another point, that people are informed about their options.
So we advise young scientists, go ahead and inform yourself. Ask for help, for money, or for everything. If you don't ask, nobody will give you. I was told like this. That's why I ask for these wonderful things to happen, and they somehow run. Exactly. But when we go back to the organoids again, I have one question that I wanted to ask you. And this is again related to a paper that I read from the lab of Sergio Pasca. You know him perhaps very well. And these guys differentiated organoids from human cells and then transplanted them to the motor cortex or somatosensory cortex of a mice. And these organoids then got integrated into the whole system, so to say, and they were functional, which is fascinating and beyond fascinating. And I'm thinking, what would be the positive side of this kind of research, for example, in your opinion?
I think there are people that they're thinking, of course, that transplantation in some cases could be advantageous. One side that you can consider, for example, could be also Parkinson patients. So you have substantia nigra. Theoretically, you're losing a lot of dopaminergic neurons. Theoretically, you can put, instead of just putting cells, you could put a small mini tissue there and see if then the cells really functionally connect and really are guided to the proper place. And theoretically, we know that with tissue engineering, you have a better retention of the cells. The cells survive better. We know this from the heart. So it could be that it's similar in the brain. It's super exciting. I have to say, I find this fascinating. I don't know if it will reach therapy. I think this is probably it's a little bit outside of my personal safe zone, so to say. I think they are much more equipped medical doctors and scientists that I'm sure they're envisioning to do this.
That's really, and we're talking about brains and heart, but of course you can differentiate livers and kidneys and basically every other organ, right?
This, I would envision that probably easier, maybe because I don't know these organs so much. But I always thought that the brain, you can do also so many things wrong. So that is what is interesting. Do the cells not form synapses and not form connections, or would you just mess up the whole thing? I don't know.
That's a good point. Good point. I don't know either. But
I guess there will be experiments where we will find out.
Yes, your PhD students will be happy. No. But there is another aspect of this organoid system that I like, and this is the drug testing, potentially. And you can use it instead of an animal model when you can screen many drugs. Of course, now there is the question and how valuable would be this information since you're testing it on a certain system. But do you test this thing? Do you have these options in your lab?
We don't do this right now, but we're setting up for this. So I think this is one of the uses that I see that organoids could have and offer a lot. So one would be drug testing, and the other point, and probably more validation, maybe not for large screens, because they are also complex systems and expensive systems, but for validating specific factors and specific medication that probably are screened with a simpler system. And the other thing is, for example, also viral for gene therapy. If you want to deliver a specific expression vector and you want to target a specific cell, which is the right promoter, which is the right particle to reach this human cell? And I think for this, such organoids can be excellent because you can really see the specificity of your vector, the specificity of your promoters, and you can use them instead of going into another model. Because in many cases, for example, rodents are very, very different. The viral vector is very different on how which viral particles actually work better for mice and human. And this could be a valuable tool to kind of bridge this gap.
And viruses you can inject in the olfactory part of the brain, and then they can penetrate the cell, the adenoviruses, for example, right?
Yeah.
You don't need to cross the blood-brain barrier. I'm not that familiar, to be honest.
So
with this.
in my case, I'm considering probably not really the brain organoid, but some other of the systems we have. But I think it is very useful from the moment that you're actually having a virus and you know it can be delivered in the organ, you want to deliver it, that you can test how specific is it, because for many of these we really don't know.
That is true. It's a great system. In this regard, I wanted to ask you something about a recent article in Science. It was, I think, last week. I will read the title and I would like to hear your opinion. So FDA no longer needs to require animal tests before human drug trials. New medicines need not be tested in animals to receive US Food and Drug Administration FDA approval, according to legislation signed by President Joe Biden in late December 2022. What do you think about this?
I will tell you my personal opinion. I came from in vivo studies, and I love organoids. I won't lie, but I think it's just another model. I think we need all the models. So organoids are great, are human, and can really give you a lot of information about the specific cell types, as I said, which can be different. So I understand the point that many of the medication fail because of the differences. But we do need in vivo studies. So whether the idea is not to use mouse studies, but actually require larger animal studies. But it's also very hard to have really the numbers. We are sitting right now in the PZ, and it's an honor to have such a facility here in Göttingen with larger animals. But it's also not very easy, and it's also super expensive. I think it's great to have organoids, but I really do think we need in vivo systems. You have then a circulation, and you have everything inside. You have an organism. And we are trying to make connections. That's why we're doing a heart muscle, and then an innervation, an autonomic nervous system, and then we would like to connect it with the brain and make a brain to heart axis in a dish. But there is still no circulation. There is currently also no hormonal regulation. There are so many things in an animal that I still cannot see that they are as useful as human organoids in my eyes. They have to be hand in hand. All models, also we learn a lot from zebrafish, and we learned a lot from so many models. So I do not think that we should just disregard the value of all the different models, but combine them and achieve the best we can. Because they all have their limitations. We have the human part. We don't have the organism part. They have the organism. They are not human. So we have to learn from all of them.
I totally agree with this view. And just a side note, DPZ is the Deutsches Primatzentrum,
Yes.
and we are here in German Primatzentrum, which is one of the biggest in Europe, I think, and it's in Göttingen. But really, I totally agree with this. As you said, we have models to model the early development, for example, with organoids, to model something else with mice. Third thing with zebrafish, we study aging with C. elegans, for example, or some other things with Drosophila even. Many people study flies to understand something about the brain. And I think every aspect is important. And I also think that this kind of a approval to bring drugs to people without testing them in animals, it's very, very dangerous. yes, I understand the point of view of the animal welfare people who want to protect animals, but I think also that we scientists put a lot of effort to protect animals, and we take care of the animals. Of course, we have to sacrifice animals very often, but still this is a very limited amount, and we cannot avoid this.
And I think maybe something that people don't realize is scientists hate that.
Exactly.
When I entered MDC, that was the first time I had to kill a mouse. I cried, and the technical assistant felt bad for me. She said, "I will kill your mice. It's okay." And I said, "No, because I'm using them for the experiment, so I felt it's hypocritical to just ask someone to kill, and then I do eventually the analysis and so on." So, but it was hard, and most of the people I know suffered. So no one is going in and saying, "Okay, great."
Yeah.
It's hard. It's very hard, and I don't think this is something we want to do. We only do it because we think it's important, because we don't want to do that with a human when you bring the drugs and you don't know what it can do.
Exactly.
So it's just, what is the other option?
I remember my first time sacrificing this animal. I had to extract the brain very fast, but I was so shaky I couldn't do it. And in this very moment, you have to be very fast and precise so that you don't damage the tissue, right? And it was very, very tricky moment. But yeah, I totally agree. I totally agree. This has to be done from somebody. Nobody likes it, but it's important. And I don't know. I don't know what is the European Union as opinion about this, whether they can bring this rule here. But let's see. Maybe the future will show. But speaking of this, I think this is a problem in science. And as you know, and we all know, there are many other problems in our community. And we talked about with Felipe last time about reproducibility. And there are also this postdoc crisis that people want to go to the industry. We have also struggles with the publishing, and we know how tricky this is. And on top of that come the grants, and how do you write them? Do you get them? You have this H index, etc., etc. What would be the biggest problem that you would like to solve in science? Maybe it's something else that I haven't mentioned yet.
It's a good question. The biggest one. So that's actually a very good question. It's hard. It's hard to not also select one. I would say that the hardest thing somehow the publication, and that you are required to have a certain amount of publications. And sometimes I experienced during my early, when I was submitting for grants early on, I got reviews that said that the grant was great, the idea was great, but I was not given the grant because of my list of publication that was not great. And I think this is putting a lot of pressure. I thought I told you I came into this very naively. I thought it would just be me observing things and writing down and understanding. And this is still what excites me about it. And of course, I want the world to know. And of course, I want people to see this and see if it is helpful. But the fact that you have to have a certain number of publications and fast if you want to be successful, I think this is putting a lot of pressure. And it's also putting a lot of pressure on young scientists. It's putting a lot of pressure on the PIs. And they also eventually put the pressure to the young scientists. So it's kind of a vicious cycle. I think the way that right now grants are granted, and the idea of if I think you can do something or not, this is what is putting the most pressure on everyone. And it could be also a reason why young scientists leave, because I say the way we have to work is I just work. And I also want to do something else besides working. And it's impossible many times to do that if you're trying to achieve this list of publications that you have to reach your dreams. So it's kind of a vicious cycle. I have to get the grant, I need the publication. To get the publication, I have to work, and then I have to work more to get the grant. I got the grant, I have to get the next publication, and you're kind of going into it, and eventually you are the lab rat and not your actual animal models. So it's tough. And this is also impacting then sometimes reproducibility, because trying to be fast, then it makes things harder. So how many times things are reproduced? I think often now, at least, it's becoming more and more important, and people are putting, it has to be X independent experiments, and now you have to do it, and it's good, or you have to test that this works in different lines and not only in one. But it's not easy, because you know that if you're not one of the first ones that publishes something, then it's very tough. Even if you have an excellent scientific piece to communicate, it's still very hard to publish it.
Yes, I agree. And I also think that this affects the studies, as you said, reproducibility, but not only. I mean, the way that we conduct studies to find out about some process, or do we do it because we want to get the grant? This is like where is the border? You want to get the grant in order to continue your study, but in order to get the grant, you have to release something, and this something is not always the best that you can do. And as you said, all over the planet, there are maybe many labs that are working on similar projects, and if they publish something before you, you're basically scooped. Although maybe this is not the correct term, but you cannot publish it anymore. And in this regard, I think, I don't know what is the solution of this problem. Definitely the granting system has to be somehow changed, but also there should be more collaboration going on with groups that work on similar topics, I think. I mean, of course, everybody has his or her ego and name, etc. It's hard, but at the end, we all know to understand the scientific truth and not to be famous. And I don't know, maybe people want to also be famous and happy and rich. Who doesn't? But yeah, I honestly don't know what is the solution of this grant and publishing problem. Do you have any suggestion?
Yeah, well, I think that at least for young scientists, the publication record should not be the definite what you need to be looking at. Probably, unfortunately, the environment where they're in should be looked at. So when you're a young scientist, you have to see in which lab they are planning to do this, what they are suggesting. So if it's doable, and it should not be only purely based in what you publish. And at the end of the day, I think we should be able also to publish negative data. And this is something I'm missing. Some of the things don't work, and these things go nowhere. Yeah, because you have no time, because you're running so many things in parallel. So it would have been great, actually, if we would be able to have also negative data and studies that show that you cannot reproduce this protocol. But this doesn't happen, right? So if we try to do a protocol and it didn't work in our hands, you cannot just say, well, we tried it and it didn't work, because you may just have done something wrong, and this will never see any publication. So somehow it's all a success story. So, and whatever is not a success is not out there. And I think we are actually typically always learning from our mistakes, and these mistakes are never published. So I think not even uploaded somewhere that it didn't work, or we did this and it proves that this is not like that. So also negative studies are very hard to publish. So I think it is a different way. I think somehow we are having too many things, trying to have a lot of success stories that they can be published, and we are working towards this. And this is just pushing more and more and more. I cannot tell you really what would be the solution. Obviously, if the grants, people have to judge somehow whether you're able to do what you're saying. Maybe There could be another way, but then if you have to examine everyone really and ask him how you plan to do this experiment, this is also a lot of administration that probably would not be possible. So I think the system was not formed like that randomly. It probably followed some kind of progression, and that was the best we could do at this point.
That's interesting.
I think in the past were less grants, and you see sometimes they have this. Yes, I grant you 10,000 euro, and the thing was, I would like to have this money to do this and this and this, and not euro, obviously. And then they were granted 10,000 for just having a sentence written. So I think things were different, of course, in the past, and now we are so many, and there are so many studies, and trying to make it more and more, it becomes more and more complex.
Maybe a bioarchive for negative data would be a nice thing. I don't know. Is there anything like that? Some website where you can upload? This is known, okay, this is only negative data. Go upload it. And if someone else is working on this and wants to check, why don't we do that? It's not that a big deal, I guess. But it will save us time, right? You don't need to try this out. Somebody else tried it and it failed.
I think the point for this is it's hard for you to put in, because if you have to write all of this, and make figures and make it feels like that's a waste of your time. But if it would be in an easier way on how you can describe, okay, this was done and this was this, and then there were some, you can upload the figures. But if you write a real manuscript, at least if you would do that, there has to be kind of, if you're not getting anything for it, it would be very hard to just, I think, to upload it. I think it's a great idea, but would people have time? Would people invest time to do it?
I think this answer to this question lies on our way how we work. So many of us have these lab notes, lab books where they handwrite things, and many of us write them on the laptop, etc. But if you kind of automate this process while you are writing your protocol or your experiment, so that you automatically generate this and this data and protocol, etc., then you upload it somewhere, then it's while you're working, you already have written this. But
maybe if we're getting electronic lab books and everybody has things like that with templates, maybe that would make it much easier. And then it's uploading the pages with the experiment description and a little paragraph on the top. I think that would help a lot.
That might
At least you would learn how people failed into doing something, right?
Yeah. These guys that are behind the cameras, they write that there is a journal of negative results, which is an interesting thing.
You see, I didn't know. We have to promote this more.
Yes. But Lenny, you want to join the talk, or? Yeah.
So apparently it's from +1, there's a collection focusing on negative, null, and inconclusive results called Positively Negative.
That's great.
Are you thinking about publishing something there?
Yes. What was it last? The CRISPR Kiko tried. No, but this is not yet. So, but yes. If we have something, I have to think about it.
See, ideas are born. That's great.
No, but this is very important.
I also agree. I mean, it is indeed important, and unfortunately, there is another aspect. Who would like to say that he failed so many times, right? For example,
who didn't fail?
Of course, but I mean, I don't care. But there are people who care about their reputation, right? Maybe I don't care because I still don't have one. Who knows?
I think we have a lot of stuff. It's just, of course, from the top of my mind, nothing comes. But I'm sure, even if I look on my own protocol books, I will find 50 things that didn't work. So I think that is important, and I think that is also important for industry. Because one of the things we are thinking, as academics, what is important for us, but I think for industry is important for them to see if a protocol works constantly and reproducibly, right? And they cannot always see that. Talking to people that they are in the industry, they tell me, well, they would have really appreciated to know if the things that they are published are working. Because eventually what you hear is, well, we tried it didn't work. Well, we also tried this protocol, it didn't work. Well, we also tried it. And at some point when you're 10 people that tried the same protocol and it didn't work, it kind of becomes a consensus. Okay, maybe it doesn't work. So
yeah, it's a good idea. I think also it's important, and we have to do it. I mean, we are the young scientists, right? So maybe we can also push a little bit the field, and who knows what happens. What I wanted to ask you also, and I see the guys are reminding me, you dropped several terms like RNA sequencing, CRISPR-Cas, optogenetics.
Oh.
Could you please briefly describe these terms in case somebody who listens is not familiar with these things?
Yes. So RNA sequencing. So it's a technique that we can actually see the complete transcriptome. So maybe a new term. So we can see all the mRNA from all the cells. And then we can tell how much of this mRNA, which eventually would be transcribed into and translated into protein, would eventually be present in our tissues. For example, if you have a specific transcript, now for noradrenergic neurons, we know we would have the gene called DBH. So if we know that we have a lot of this mRNA, then we know that our organoid is really turning into the proper fate. And then we are using such tools to understand if you're doing a bulk RNA sequencing, means we take all the RNA, and then we are just seeing in this pool how much of these transcripts are in. And we are looking at the abundance and say, well, if I have a lot of DBH, probably I have a lot of noradrenergic neurons. And if you're looking at single cell sequencing, then you're really isolating cells or nuclei when you're doing a nuclei sequencing. And then you can see what is the expression in these individual cells that you isolated, and you can cluster them, and then you can see really I have these populations more in an organoid or this population more inside. So that was the one term I mentioned. The second term I mentioned was optogenetics.
Optogenetics, CRISPR-Cas.
Yes. CRISPR is a technique we are actually using to integrate, to genome engineer. So we are changing the genome by using target little gRNAs. And what we are doing is we can integrate several targets. And one of these targets, for example, could be an optogenetic. So it can be a channel that is activated by light, and it allows then the neuron to depolarize. And this allows us to really stimulate neurons by just shining light on them. And we're using this actually to specifically then target the neurons. You can use specific promoters like synapsin, and then you can control the activation of the neuron and not of the cardiomyocyte or another kind of cell. So, and CRISPR, I think I mentioned, right? And optogenetic, I think that's very broadly, but
very broadly.
And you can use CRISPR, of course, to knock out specific genes or even to introduce mutations. So you can just remove, let's say, the ATG, the transcription start site, and then you stop the protein of being the gene or transcript. And then you can also knock out or knock in a specific mutation, which you know, for example, that a patient has. So nowadays, you could have the patient and you could correct it and see really if the mutation is responsible for the disease, or you could just have a cell line which is a wild type that you're working in, and you say, well, I want to see what this mutation is doing. And then you can make this mutation into the genome. And then you can make disease modeling.
That is great. I mean, these are fairly new technologies, and we use them regularly in Göttingen, for example. But it's a little bit tricky with CRISPR-Cas and neurons, right? They're post-mitotic cells. I mean, in terms of organoids, perhaps you have still this stem cell proliferation and division, but when we want to apply it in adult animals, then it's a little bit harder because these cells don't divide. Exactly. I struggled with this at some point, but I think people are working on solving also this aspect to some extent. There are different techniques, the I forgot the name, to knock in TKIT, CRISPR, when you cut on both sides and you integrate, it's flop the gene. Anyhow, it doesn't work.
Orange I have to CRISPR, do some reading.
Yeah.
I didn't hear this before.
I mean, it kind of works in some neurons, but not always. It depends on what you want to insert, etc.
We don't do this in neurons. So we are doing everything in stem cells and use pluripotent stem cells, which I also didn't introduce, right? I said what stem cells is, but I didn't say what is induced.
Exactly.
Did I say?
We said that we can reprogram them.
Yes. Then yes, I mentioned it. You're right.
Cardiomyocytes are interesting cells, but you haven't talked about them so far, or you mentioned them briefly, but you haven't explained what are these guys doing.
Yes.
Right.
They are actually the electrically excitable cells of the heart, and they are the ones that they are responsible for the pumping function of the heart. They are contractile cells, and they are responsible for the blood to be pumped out or going into the lungs and then going into the body. And we are very interested in these cells because obviously if they are dysfunction, their dysfunction is leading to cardiac dysfunction and in many cases, of course, to heart failure. And of course, if they are supposed to be having a rhythm, so they are for the heart to pump the blood, you have to have a specific rhythm that works, that it's organizing the atria and the ventricles and how the blood then is circulating. And for this, you have actually the conduction system, and the conduction system is with the cells that actually also the autonomic neurons really are really close to. And this is what is giving the electrical pulse in the heart. And you need this thing to be organized, not to have arrhythmias. So you need to have a conduction system that is initiating a pulse, and then you have that all of these cardiomyocytes then are connected to each other, and then you can have a proper contraction.
I would like to ask you 2 or 3 final things. So the first one is very easy and the second one are not that easy.
Oh.
How the hell do you keep with literature that is out there?
I don't know. So I don't think I know all the literature out there. It's impossible. It's impossible. I think nowadays, probably before going to sleep, you're just looking through Twitter and you see a lot of things coming up, and some are interesting, some not for what we're doing. Some actually almost subscribed in all of the publications and so on, and a lot of organoid stuff, and at the end of the day, it feels like, to me at least, I learned the first things through Twitter, and then you get PubMed alerts. So it feels a bit like that. You have to read a lot, and reading becomes more, for me, it's like before going to sleep, probably. So I also have a big neuroscience book that I was reading because I'm not a neuroscientist. I just love neurons.
Which one do you have? The Candel one?
Candel,
yeah,
yes.
Of course. Okay.
And I had it, and my son was laughing, and my daughter brought Harry Potter, and she was like, "Okay, Mom, look, my book is also as big." I was like, and I said, "I hope it's as fun as mine is." Actually, it is hard. It is hard, and also not only about reading what is out there new, but if you're changing the field, you have to enter, and going, I think, into the brain, it was amazing, but it's really so much fun. It's so much complexity that I still feel like I know nothing. So it's really fun. For publications, you have to read all the time, and as I said, it's a similar. So for me, it is an evening read, so something interesting, and
I do that rather in the morning. Before I check emails, kind of over the literature, what is new in Nature, Science, Cell, Twitter, any other journal. I have some favorites there, and the rest is, friends are telling you or something.
Yes.
But rather in the evening, I'm more, let's read some other book, nothing related to science, so that I can sleep easily.
Yeah, wait a little. I was also you.
Okay,
Some time ago.
This is a nice transition to the next question. What is your one advice that you would give to a PhD student starting now?
I would say that they really should follow their dream. They really should follow it. If they really love what they're doing and they really like what they're doing, they should follow it. And if they feel they are not really thrilled, they should leave. Mm-hmm. I would think that looking back, I would have stopped doing the PhD I was doing at the time. I should have stopped. I should have done what I felt like I should have done was to leave. I felt at the time, I'm 1.5 years inside. It's okay. But I was not excited. I was not that excited as much as I was later on with the science I did. And I think you should when you are doing something, you should be really excited about it. It doesn't matter what it is. It can be in academia, it can be in the industry, it can be wherever it is, but it should make you happy. Mm-hmm. It will be a struggle. I'm not saying that it will just be flowers but it should still be stimulating. And if it's not stimulating, then it's not worth it.
Yeah. For me, I mentioned it, I think, last time, the idea that we are people who... So we know that amount of information, and because of our work, we will know this amount of information. And slowly we will build up some knowledge, and we are kind of the people that push the progress in a certain domain, right? Of course, there are other scientists, business people, sport, art, everything is important. But for me, the creation, so to say, of knowledge is something that is, wow, okay, why not? And keeps me going. But let's see how long, right? I'm still in my early scientific development. Yes. And the last question, of course, if you want to add something, feel free, but I will not let you go before this one. What is the one thing that you would like to solve in terms of neuroscience with your research?
I would really like to understand how arrhythmias are happening due to neuro and cardiomyocyte crosstalk. Or I would really like to know if a diseased cardiomyocyte, what does it tell a neuron? And what does a diseased neuron tell a cardiomyocyte? Because the cells are not just there and throwing neurotransmitters to each other. And they are not just, yes, I give you something, you respond. We know that there has to be another kind of interaction. So I would really like to understand, and not only cardiomyocytes, there is also vasculature. There are a lot of cells in the heart, and a lot of people are looking at these cells, but we kind of forget the neurons. And for me, this is very interesting. I have a lot of things, and maybe if you ask me in 10 years, I will have a completely different question. But right now, from this trip that I have done scientifically so far, this was one of my initial questions. What is this interaction? How do they interact? And how do they interact in a healthy tissue? And how do they interact in disease? That I think it is one of the things that I would really like to know.
That sounds great, and I wish you a lot of luck and success for the future, and I hope that you succeed in this domain, and maybe you find about other exciting things that will keep you going, I don't know, until the end. And yeah, I think we guys are doing great so far, and thank you very much for being here. I hope we will meet around in Göttingen or in the MBXC retreat, or I'm actually coming to your lab in April for a method course. Exactly. For the 2 days.
Organoid?
Yeah, the organoid.
Very good.
So yeah, I'm excited about it, and thank you very much once again for being here.
Thank you very much for having me.
Neuroscience and Beyond, Know more, get inspired.